In this video, I'll demonstrate how to use PACUPP to identify and visualize cavities and pockets in atomic resolution 3D models of proteins and other macromolecules. This is the enzyme acetylcholinesterase. PACUPP has filled its catalytic cavity with pseudoatoms, which are the colored atoms. Inside the catalytic cavity, at the bottom, you see the inhibitor huperzine A. In this video, I will show you how to use PACUPP and also some other examples of cavities revealed by PACUPP. Here's what you get when you download PACUPP. Aside from the instructions in the folder 0 Instructions, the zeros to make it be at the top of the list, the most important things are the 1, 2, 3. 1. Double-click the Jmol.jar file to run the Jmol application. 2. Load a macromolecule, and you can read this PDF document for lots of details about different options for doing that. 3. Drag the Fill Cavities script and drop it into Jmol to start the PACUPP job. Here are the contents of the remainder of this video, with starting at points so you can jump directly to parts that may be of particular interest to you. Before you explore the cavities in your macromolecule, it's a good idea to be familiar with its structure. First Glance in Jmol is an excellent way to do this. First Glance tells you a lot about your protein in plain terms and without needing any command language. Yet it offers plenty of power and depth, even for researchers. We'll start with HIV Protease, 1-IZH. Notice that there are two protein chains, a green chain and a blue chain. Over here in the left panel, it says there are two chains. The equal sign between A and B, the names of the two chains, means that they are sequence identical. So this is a homodimer. These space-filling atoms between the two protein chain ribbons are a ligand, namely an inhibitor of this HIV protease. This type of inhibitor is what has saved so many lives of HIV-positive people since the late 1990s, when these inhibitors were first approved by the FDA. Now we'll go ahead and start the Jmol application. The first time you open Jmol by double-clicking on the jmol.jar file, this is what you will see. But you're also going to need another window called the Jmol script console. To get that, click on File, then go down to Console, click on Console, and this white window opens here. Notice it says Jmol script console at the top. You can resize these windows and position them anywhere you want, and a very handy feature of Jmol is that if you open the File menu and exit from Jmol by this means, it will remember exactly where your windows are, which screen they're on, their sizes and positions. The next thing is to load HIV Protease into Jmol. I'll type load, L-O-A-D, space, equals, no space, 1-IZH, press enter, and the HIV protease model has come through the internet from the RCSB Protein Data Bank. Now we'll go to our PACUPP download, grab the Fill Cavities script, drag it and drop it onto the molecule, not into the script console, onto the molecule. PACUPP is now asking us whether we want to use the default settings, which are best for fine definition of small cavities. If it's the first time you've looked at a molecule or you're not sure, just say yes, and then you can make adjustments in later jobs if necessary. So we'll click on yes. PACUPP is now running. As it runs, it reports what it's doing in the script console. This will probably go by too fast to read, but that's okay. You don't need to look at it. It's there, however, if you want to scroll back up later and take a look. On the right in the script console, the very last thing PACUPP reports at the bottom is how long the job took. In this case, 6 seconds. It's a very small molecule. And just above that, you'll see a description of PACUPP's initial display, followed by some commands that you might want to use. The opaque colored clusters of atoms are the pseudoatoms, yellow in here, green, purple. Each cluster is a different color to help you distinguish them, and each represents a pocket or cavity of some sort. You'll notice that the pink, green, and purple ones are shallow pockets on the surface, but the yellow one penetrates right through the center of the protein. So we suspect that this is where our inhibitor is, and this is the catalytic site that we saw in first glance. So I'm going to click on that yellow cluster, and that isolates it. That's a toggle. If I click again, all the other cavities would come back. The light gray translucent atoms are the protein, and we can hide those by entering the command M for macromolecule. This is also a toggle. If we entered M again, it would come back. Now, we're pretty sure this is the catalytic site cavity, but to verify that, we'll enter the command R for residue, and then we can type in the known catalytic residues aspartate 25. And you see they appeared here as sticks right next to the yellow cavity. So that confirms that this is the catalytic region of the molecule. Now we'd like to have a look at this catalytic region of the molecule in more detail. I'm using my mouse wheel here to zoom. The last thing that PACUPP suggested in its report was that we type the command V for views. So I'm going to do that now. And we're offered a whole series of options. The initial view, we can see what's inside cavities, what's clashing with the pseudoatoms, the macromolecule atoms that line the cavity, cavity linings, the mouths of the cavities, and we can measure a cavity. So we'll take a look at what's inside this cavity, and we see the inhibitor inside the yellow pseudoatoms, along with a couple of water oxygens right up here, which are not really in the cavity. They're in this little extension that's actually a shallow groove on the surface that happened to be connected to this cavity. Next, we might like to know what atoms of the protein are forming the boundary of this cavity, that is, lining the cavity. So in order to do that, we'll enter V for views again, choose cavity linings, and now we have a choice between polar lining atoms, apolar, water, and other, which would be metals and other unusual elements. I'm choosing polar, and now we see all the oxygens and nitrogens that are within 5 angstroms of these pseudoatoms. I'm going to click on this oxygen atom, and a report over here says it's aspartic sequence number 30 in chain B, and I clicked on the oxygen delta, the fourth level out from the alpha carbon of that aspartic acid. I'm entering the command L to list the lining atoms. Here they are, and they're not only displayed in the Jmol script console, but that command L also writes out these atoms in a spreadsheet ready file. You can drop the file right onto Excel and have all those atoms in a spreadsheet. Let's take a look at the apolar atoms that are lining the cavity. So entering V again, cavity linings, and apolar. Here we have all the carbon atoms within 5 angstroms of the pseudoatoms. This would also include sulfurs, which are generally apolar and protein. Now let's take a look at the cavity mouths. So entering V again, cavity mouths. But before we do that, let's go back to the initial view so that we'll see all the cavities. That's a more useful view of cavity mouths. So here we see all the cavities. And now cavity mouths. The protein is dark, so the entrances to the cavities stand out. You see the yellow cavity does not show through this part of the protein, but you can see the 2 entrances to the yellow cavity. Now I'm returning to the initial view. And let's measure this yellow cavity. What are its dimensions? So I'll enter V again and choose Measure Cavities. This shows the cavity as translucent, which helps in the measurement process. So what about the length, the longest dimension? I'll double-click on that atom, and as I pull my mouse away, distances to other atoms are shown along this dotted line. Double-clicking again on this atom tells us that there are 16.4 angstroms between the centers of those 2 atoms. The outside dimensions would require that you add 1 pseudoatom diameter, which is 3 angstroms in this case. You can see that in the report from the PACUPP job. Let's measure a distance across. Double-click here, double-click here, and we have 8.5 angstroms between those atom centers. So that is the cavity measurement process. We've seen how to use PACUPP on a straightforward example. Now let's look at a few completed PACUPP jobs, starting with the DNA double helix B-form DNA. Here you can clearly see the major and minor grooves. Here we have, in first glance in Jmol, our next example. This is the spike protein of SARS coronavirus 2, the cause of the COVID epidemic. This protein is essential for infection by coronavirus. It is a trimer, 3 protein chains. And This is the side that would be bound to the membrane, but the membrane binding part was not present in the experimental protein that produced this model by electron microscopy. This is the side that binds to the angiotensin converting enzyme 2 receptor for entering the host cell. There are 3 chains, and as you can see, A equals B equals C, meaning they are sequence identical. If we click on chain details, it confirms 3 sequence identical chains. Now we're back in the Jmol application, looking at the PACUPP result for this coronavirus spike protein, and at the bottom you can see a small cavity. This is being researched as a possible drug target to prevent membrane fusion and thereby prevent infection by coronavirus. The cavity, as you see it here, was using a cavity detail level of coarse, which makes a rather simplified, almost schematic version of the cavity. Next, we'll look at what happens when we specify a cavity detail of fine. Here is the result of a PACUPP run set for fine cavity detail. You see that the surface is covered with shallow pockets, and so was it, by the way, with coarse detail, but I had hidden those other cavities before I showed you the one of interest. I'm going to hide the translucent space-filling protein with the command M for macromolecule. And at the bottom of this is the yellow cavity, which is the one of interest. So I'm going to click on that to isolate it, and then zoom in with my mouse wheel. And now I'm going to color the pseudoatoms by depth. The command for that is D. Now the red atoms are atoms that are exposed to the outside. They are entrances to this cavity. As the color moves towards green and blue through yellow, you're getting deeper and deeper into the protein. You see that these entrances through the red regions are rather thin, and when the cavity detail was set to coarse, they were not reported because those thin entrances were not detected by the coarse probe. This region here, the deepest part, is the part of interest, and these may be called fenestrations, or entrances to the cavity. For our next example, we'll look at a proteasome. This is a huge structure. It took 5 minutes for PACUPP to process this. But PACUPP has a batch capability, so you can set up a large molecule like this to run through multiple sets of parameters unattended. It has 28 chains, although they're only in 2 distinct sequences. If we look at one end of this structure, we can see that there is a hole all the way through the structure. We want to know the shape of that cavity. In First Glance in Jmol, as we are now, we can go to the Views tab and push the Slab button, and that cuts through the center, and we see that it almost looks like 3 separate cavities here. If we stop hiding the back of this, it allows us to look with just the front half removed. We sort of get an impression of the shape, but we'll get a much better impression with PACUPP. Here is our result. PACUPP, job with the proteasome 1 PMA. Because it's such a large structure, instead of using a fine level of cavity detail, I set it to moderate. and I set the cavity size to medium instead of small. So what do we have at the end here? I will click on it to isolate this region, this group of pseudoatoms. I'm going to hide using the M command here to hide the translucent macromolecule. And you see that what we isolated is just one of the three chambers in this proteasome. Clicking it again, and clicking as a toggle, they all come back. I'll click at this end, and again we have just one end. What about the middle? You can display multiple selected cavities in PACUPP by getting the cavity ID numbers. So we see over here that one of these is cavity 43. The first one. The second one was cavity 13. What is the middle one? Click to restore all cavities. I'm having trouble hitting the pseudoatom. I keep hitting protein. And we'll use a command S, which means slice or slab. It's a toggle also. Now we're seeing a thin slice through the middle. We know that this is cavity 13. This is cavity 43. And this is cavity 24. The command for this is CAV space. First I turned off slab. CAV space, and then the three numbers, 13, 24, and 43. Enter. And now just those three are displayed. Very pretty. They're colored individually because they're not actually touching each other. This is what PACUPP calls the group cavity coloring scheme, the group color scheme. Each discrete group is given a different color. They're hollow, so what that means is we need to increase the cavity size from medium to large. So we'll do that next. So here we have the PACUPP result for the proteasome 1-PMA with a large cavity size. That's the one change from the previous job. And let's click on the end and see what we have. Isn't that beautiful? We'll enter M to hide the translucent protein, leaving the rainbow-colored backbone traces. So all three chambers are now touching each other, that is, the pseudoatoms are, and so they get a single color in the PACUPP group color scheme. Entering D to color by depth, we see the entrances here and here, and as we move farther in from the entrance, we go through the spectrum towards blue. These pseudoatoms are the deepest, and if we use the S command to slice this, we see a very pretty picture, and now these are not hollow. And that means that the volume estimate is reasonable, and it's about 150,000 cubic angstroms for this entire channel. We can get a really good look at the shapes of these chambers now. I just will touch briefly on this example without going into detail. This is the 50S portion of a ribosome. It required some manual Jmol scripting to get this view, the details of which are in a slideshow that you'll find at the PACUPP website. This is the transfer RNA. This is the nascent peptide in black being synthesized off of the tRNA. And here are the pseudoatoms that PACUPP has inserted to represent the nascent peptide exit channel of the ribosome. There is the exit in red. RNA is in red and protein is in green. And you can get all the details, as I mentioned, the technical, including the scripts, in the slideshow that you'll find at the PACUPP website. For our last example, we'll look at acetylcholinesterase because it illustrates some additional capabilities of PACUPP that can be very useful. The first thing that PACUPP tells us in this particular case is that this molecule has a biological assembly that is different from the asymmetric unit, and it suggests we might prefer to be working with the biological assembly. If you click Cancel to get BA, biological assembly, it will give you a URL to get a very practical form of the biological assembly, which is more practical than what you'll get from the protein data bank. But I happen to know that in this case, I'm happy to continue without using the biological assembly, which is a dimer. I just want to look at one copy of the chain here, so I will click Continue without biological assembly. In most cases, especially if it's the first time you're running PACUPP on a molecule, you can simply accept Yes here to use the default settings. In PACUPP terminology, the default settings are for fine definition of cavity detail and for cavity size of small. But in this case, previous PACUPP runs have shown me that I want extra fine definition of small cavities. So I'm going to click No here. That will allow me to customize the settings. By default, the macromolecule will be outside cavities, defining the cavity boundaries, and water or ligands, that is heteroatoms in PDB terminology, will be inside cavities. This dialog offers you the opportunity to specify that something unusual should be inside cavities. For example, if there's a peptide ligand, since it's protein, it would by default be outside, but if it's chain B, you could say here chain equals B, and that would put it inside cavities. In this case, we don't have anything unusual to specify here, so I'll just click OK, leaving it blank. The same thing for any special atoms we want to designate to be outside cavities. This dialog asks us to specify a level of cavity detail. As I mentioned earlier, I've already decided that I want extra fine detail in this somewhat unusual case. This option to offset the 3D grid of pseudoatoms is rather technical, and we just don't need to get into it now. Next, PACUPP asks us what cavity size is appropriate. Earlier, you saw that for the proteasome, we needed a large cavity size, but small is perfectly fine in this case. Now, PACUPP has started filling the cavities in this protein, and it's reporting its progress as it goes along in the script console on the right. In most cases, these reports will go by too fast to read, but they're there if you want to scroll back later and take a look at them. Also, all the specifics of each job are written into a report file. Now PACUPP has finished, and you can see at the very bottom on the right that this job took 28 seconds, which is about 3 times as long as it would have taken if we had specified fine cavity detail instead of extra fine. There will always be some very small cavities that contain only one or a few pseudoatoms each, and these just amount to clutter, and we don't need to see them. So by default, PACUPP shows you only cavities with 5 or more pseudoatoms. In this case, as you'll see right here under the initial display title, PACUPP is showing us 25 cavities that have at least 5 pseudoatoms each. I'm pretty sure the cavities I'm interested in will have many more than 5 pseudoatoms, so I'm going to hide some of those smaller cavities. To do that, I'm typing in the command cavmin, C-A-V-M-I-N, which tells me what its possible values are. I'm going to try cavmin 10. This shows me the 11 cavities that have 10 or more pseudoatoms each. Now I'm trying cavmin 25, and we're down to 6 cavities. I think that sounds good. So the next thing I'm going to do is color the cavities by depth from the surface. To do that, I enter the command D. What I'm looking for here is the deepest cavity. The deepest cavity will have blue atoms at its greatest depth, and perhaps you can see it already. But in order to make it easier to see, we need to hide the space-filling macromolecule, which is in light gray. The command for that is M. It's a toggle. And now we have just the backbone trace. It's easier to see that deep cavity, but I'm going to enter B, which toggles off the backbone trace. And now it's real clear where our cavity of interest is. By simply clicking on it, we can isolate it. So here I'm going to click on the blue atoms, and the cavity is isolated. Clicking again on the isolated cavity restores all of the cavities. So this is a toggle. Our cavity has a couple of wings sticking out from the sides. So what I'm going to demonstrate next is 2 methods that PACUPP offers to trim off these wings. They're not really part of the main cavity. They're just shallow surface depressions here connected by a single atom by accident, and you may not wish to show them, as I didn't in the very early view I showed of acetylcholinesterase in this video. So to start with the first method, we'll use the V for views command and go back to the initial view. And now I'll hide the macromolecule, hide the backbone of the macromolecule, B, and color by depth. What we're going to do now is peel away the outer layers of pseudoatoms. The command P peels off the red atoms, and you can repeat this as many times as you wish, peeling more and more outer layers away. Now we've got just some blue and light blue, but there's one stray atom here I want to get rid of, so one more peeling cycle. These purple clumps over here are buried cavities that have no connection to the surface. They can be hidden with a special command BU, 2 characters, for buried. And now we have just the tip of our cavity of interest. Now what we're going to do is extend it out with the command E for extend. But this command is a little peculiar. It adds only the pseudoatoms in the next layer, the next outer layer, that are in contact with already visible pseudoatoms. This means it doesn't actually recreate the entire cavity. Here it says the outermost layer is already visible, so we've extended as far as we can. But now what we need to do is fill out the cavity with a few other atoms that didn't meet those criteria. The command for that is F for fatten. We're going to fatten the cavity through a few cycles until it has nothing more to fatten. And here it says it's shown us all the fattening it can. The fatten command does not go out to layer 1. So if we want to add layer 1, the red layer, we need to use the F1 command, which fattens layer 1. I will just do one cycle of this, and that is a very nice representation of the complete cavity that we wanted. So by peeling and fattening and extending, you can often get exactly what you want with no direct surgery, so to speak. Next, I'm going to show you the surgical method. This is cavity 67, as you see. So by entering CAV 67, that will show just this one cavity in its complete form with its wings. And now we're going to use the surgical method to remove these wings. The first command is A for atom. It allows you to hide one atom at a time by clicking. This atom is connecting this wing through a single contact. There, we've hidden it. The other one is a little better connected, but it's really only 3 clicks to disconnect it. So there's one. There are 2 atoms here that need to be deleted. By the way, if you make a mistake, if you click the wrong atom, enter the command U, and it will undo your latest click. There we go. Now that wing is disconnected, and we're ready to proceed with major surgery. So for major surgery, we need to be able to hide entire unwanted clusters with clicking. The command for that is K. So each click will now hide an entire cluster of contiguous pseudoatoms. So goodbye to you, and goodbye to you. If I should accidentally click my last remaining cluster, the K command is turned off automatically rather than hiding the last cluster. But if you should inadvertently do something disastrous, don't overlook this undo button down here. It can be a lifesaver. And also don't overlook the font size button. By clicking this, you can change the font size in the Jmol script console, going through a whole series like so. Now that we have surgically cleaned up the catalytic cavity so nicely, let's see what's inside it. So entering V, we can click inside cavities, and there's huperzine A, the inhibitor, and a couple of waters. If we want to click on the atoms inside the cavity to identify them, we need to hide the pseudoatoms because they're covering up the enclosed atoms. So the command for that is cavtoggle, or CT for short, and that simply temporarily hides the pseudoatoms, allowing us to click on the contents of the cavity and identify HUP to find out what that means. Go back to first glance. It tells you what each ligand is, its full chemical name. This is water. And then entering CT again, we bring back our nicely cleaned up cavity. And to show it in context, we might enter B for backbone, zoom out a bit, or if we prefer, we can turn off the B, turn on the M for macromolecule and get the gray translucent version. And this, in fact, is the scene that I showed at the very beginning of the video. You may be wondering how on earth you're going to remember all these commands. Well, you can simply enter H for help, and PACUPP will display all of its commands in a rather verbose, detailed list. Here I've gotten up to the top where it tells you enter H or cav help or CH. Here the commands are grouped by categories with considerable explanation. For a shorter list, I'm going to first hit the clear button at the bottom of the script console and type AL for the alphabetic list. It's the only other list of commands. Here they are alphabetically. So you might want to print this list and have it handy when you're working with PACUPP to help you think of which command would be appropriate for what you want to do. Of course, the PACUPP download includes both lists of commands as PDF files, which makes it easy to print either one. PACUPP is a Jmol script. It fills any spaces between the atoms of a macromolecule with pseudoatoms. This reveals pockets, cavities, tunnels, and channels in a protein or other macromolecular complex. As you will see, PACUPP provides many tools to help you visualize and analyze the cavities that it reveals. PACUPP creates a new PDB file to which it has added the pseudoatoms that fill the cavities. These new PDB files are especially useful for very large molecules which may take several minutes to run in PACUPP. You can drop the new PDB file into Jmol and have all of PACUPP's capabilities available without rerunning the job. These new PDB files can also be used to look at PACUPP's results in software of your choice, such as PyMol, Chimera, CN3D, Visual Molecular Dynamics, etc. Jmol is a Java-based application. That means it works equally well in Windows, Mac OS, and Linux, and therefore so does PACUPP. Down below this video, you'll find a link to the PACUPP website where you can download PACUPP, see a slideshow with animated sample results, get the complete documentation on how to use PACUPP, complete command reference, and so forth.